Back to we're live now. So.
Yeah, okay. Hello, everyone. Good evening. It's wonderful to see you all here. Thanks for coming out. This is a great venue. It feels a bit like a comedy club, brick wall and all. Yeah, welcome to the Café Scientifique. Pardon my French. Yeah, of course, please. Thank you. My name is Peter Log. I'm the Dean of the Faculty of Science, and it is a real pleasure to welcome and introduce Jen Cobb here. I can take that. Jennifer Cobb is a professor in biochemistry and microbiology, a reasonably recent addition to the University of Victoria. Jen got a graduate degree, a PhD at Tennessee, and did some postdoctoral work in Switzerland, in Geneva. And then spent 15 years in Calgary, and then we were able to recruit her to come to UVic. Her talk today is The Fountain of Youth. Yeah, I figured. Well, you know, there's some elixirs in front of most of you, so maybe that will do. Subtitle, Longevity, Health Span, and Cellular Aging. So please join me in welcoming Jennifer.
Thanks very much, Peter. So aging's pretty interesting, huh? We're all doing it. It happens at the level of the organism. It also happens at the level of your cells. And those two are related, right? So tonight, we're going to be talking about biological aging. And the definition that is commonly used in journals and scientific literature on aging is that aging is the time-dependent decline of function, okay? And so it's pretty broad, but it applies to what's going on at the level of the organism and what's going on at the level of the cell. So the good news is we are living longer as humans. The reason for that is not because we've discovered the fountain of youth yet. It does have to do with medical advances and antibiotics, vaccinations, sanitation, those kinds of things. So there's less death due to that than there are to what are age-related diseases, which are due to physiological changes inside our body. So Andy Rooney said, everybody likes the idea of growing or of living a long life, but nobody likes the idea of aging, right? So within the fountain of youth, really it's our aim to try to slow that decline of function as much as possible, and if we can, reverse it to any certain degree. So as I said, we're living a lot longer, and the number of individuals over the age, so in Canada, the number of individuals over the age of 65 is greater than the number of individuals under the age of 14. And that's been true since 2016, and it's only that gap will only become wider. We're living twice as long as we did in 1900. So the average expected lifespan in 1900 was 41. Today, it's 84. Again, this is due to advances in antibiotics and so forth. So what is limiting it now? What's limiting our lifespan now? Well, people are dying of age-related diseases such as cardiovascular disease, cancer, neurodegenerative diseases. Those are the big three. And they have a cellular component too. So when individuals with these diseases present, in the clinic with this disease, there's a disease at the cellular level too. So there's disease cells causing these diseases. And so, when life science research, a lot of funding and resources and people who are interested in these diseases, they've been at the front burner of life science research because they've taken a number of lives, right? Aging, on the other hand, as well, has a cellular component. That's kind of been more back burner. You hear in the scientific community, or had, it just happens to everybody. Well, that's true. So, the diseases have gotten a lot more attention than, the study of aging. But you get these as you get older. Nonetheless, aging researchers they've still progressed with their research. And about 10 years ago, there was a compilation of all the characteristics that are associated with aging. And it was coined the hallmarks of aging. And what became quite clear is that many of the pathways that are disrupted in aging are the same ones that are disrupted in these diseases. So there's a lot of overlap here. And if we can figure out what's going on in aging, it might have implications for what's going on in all the age-related diseases. So one area or one hallmark of aging, which our lab has been very interested in, is understanding the relationship between genome instability and aging. And this is a big driver of aging. So what is genome instability? Well, first I'll tell you what genome stability is. So in a cell, you have a nucleus that houses your genome. So the genome is, the beautiful duplex DNA that is basically the blueprint of life. And it's not, just in there like a bunch of spaghetti. It's quite organized. So it's in the nature's alphabet of, A, T, G, C. It's organized such that information that's needed can be fairly easily accessible. And when all of that's running perfect, you have a stable genome. An unstable genome is when things get a little shuffled. And one of the big drivers of genome instability, or messing this all up, is DNA damage. So there's many different forms of DNA damage. You can have, breaks in single-stranded, double-stranded bases. There's many different types of DNA damage. How it's repaired, how long it persists, how it's repaired, and what the chromatin looks like. So the DNA is wrapped in chromatin with proteins, and those are all, how it's packaged is also considered part, I'm considering that part of genome stability. So if there's high levels of DNA damage, normally repair factors go to those sites. So on average, there's 10 to 100,000 lesions in every cell of your body every day. That's a lot of damage, a lot of damage. Luckily, our cells have normally highly efficient repair enzymes. It's not always perfect though. And one area that we've been focused on is really trying to understand during the normal aging process how aging changes in an old cell compared to a young cell. What's different? When repair isn't as functionally efficient as it should be, this can lead to mutations that accumulate in your DNA, in your genome. And it's known that individuals or cells derived from older individuals have a higher mutational burden and have higher levels of DNA damage. In addition to that, even if the damage is repaired, the chromatin environment can be somewhat changed. So think about the genome, the blueprint. Okay, so if one of the A, T, Gs, or Cs is messed up, repair factors come and put that back together. But when I say the genome is organized, it also is highlighted. You can highlight the blueprint in areas. You can put little sticky tags on, this is the area, these are the genes that need to express under these conditions. If there's damage in those areas, after repair, sometimes, the sticky notes aren't there anymore, or the highlighters faded a little bit. It's not functioning as it should. So, given that genome instability is a hallmark of aging, what are some of the correlations, evidence in the field that really supports this? And so this has been done, I'm kind of giving a general overview. It's both due to individuals, so you can look at it on a number of levels, and there's a number of ways, evidence to support this. One is, so we were talking about cancer as being an age-related disease. There are also children that get certain types of cancer. They tend to be very different than those that adults get. But individuals that have childhood cancer, that have received chemotherapy or a lot of genotoxic agents in their treatment, they age much faster, and both at the level of frailty. So individuals over 65, there's a certain percentage of those individuals, I think it's 10 to 15, that mark as frail. Individuals that have received chemotherapy in childhood, they're at the same rates as the over 65 group at the age of 30. By the age of 50, these individuals are much more likely to get a secondary kind of cancer. You know, you might say, well, maybe they have something, you know, in their DNA that allows this. But it's not just cancer that they succumb to. They're also at a much higher rate of cardiovascular disease and neurodegeneration, so other types of aging. Probably one of the best evidences for really demonstrating this relationship between increased DNA damage overall and aging is what are called progeria syndromes. So these are syndromes that are premature aging syndromes. And there's quite a few of them. They can range from very fast aging, like they can present at age 2, and those individuals will die in their teens to 20s. Some of those progeria syndromes, you know, individuals don't know that they have this until they go through puberty and they fail to kind of enjoy the growth spurt, and then they dramatically age. And all of the factors that are associated with progeria syndromes are all factors that are involved in DNA damage and repair and genome maintenance. And these individuals, this is a germline mutation. They're born with this. And this is really an elegant yet tragic demonstration of how important DNA damage repair, you know, mechanisms are in human health. So if you don't age, if you don't have, you know, the correct machinery for repairing your genome at birth, you know, this leads to premature aging. Early in cell division, the number of times a cell divides, repair level efficiency goes down, you accumulate damage. So that's in that direction. But it doesn't really, you know, that's correlation, causation, but kind of, you know, is there a gold standard? What if, can you increase repair, or can you upregulate DNA damage and repair, and, you know, does this help ameliorate some of the aging hallmarks? It's kind of hard to do. There's no known compound that increases DNA damage repair per se. It's known that some compounds, if you look then in the genome, they have less DNA damage, but it's not clear if that's actually coming from increasing DNA damage repair factors. And because there's so many of them, you can't just like overexpress one or make one good, because they kind of, you know, they work as a group. Some evidence suggests, though, both primarily in the lab in model organisms, like using C. elegans. So these are little nematode worms that genetic studies have been done on. Screens done using those model organisms, labs that work in that system, what they did is they looked for worms that were long living, and then they went back and found what mutations correlated with that. And they weren't, you know, some were DNA damage and repair, but overall, what was common was that those worms were more resistant to genotoxic stress. It could be that, you know, it wasn't being transmitted the way that it was in a normal worm, you know, so the cell actually saw less oxidative damage. So as damage builds, and you get a lot of it in your cells, how is that then related to aging? Well, it's really complex, and I'm just going to focus on the damage part, but there's a lot of mechanisms in the cell working. But one thing is known is that if you have a cell and it's bombarded with damage, it's going through cell division, it's accumulating damage, one of the things that happens is the cell recognizes, oh, I've got a lot of mutations, I've got a lot of DNA damage, I shouldn't divide because this could be bad news. So they kind of put the brake on things. And at that point, there's two outcomes. One is apoptosis, or programmed cell death. The other pathway is called cellular senescence. And that means that the cell stops dividing, but it doesn't die. Cellular senescence is all of our cells, they have to make this decision at some point if they're not going to divide anymore. Cellular senescence can be triggered, and this is maybe where you maybe have heard more about the connection between genome integrity and aging, is at the telomeres, right? So the telomeres at the end of your chromosomes, many times when this is talked about, it's like a shoelace, and the telomeres are like the little plastic ends on the bottom. And every time a cell goes through a division, it replicates its DNA, the cells divide. But the way replication proceeds, the telomeres get shorter with every cell division. So at some point, that little plastic end isn't there anymore, and the shoelace frays. That causes cellular senescence. Cellular senescence is also triggered when the middle of that shoelace maybe is cut in half due to damage, or it's got bleach on it from oxidizing agents. So DNA damage leads to cellular senescence, as does telomere attrition. Mitochondrial dysfunction also leads to cellular senescence. And what happens then is that when a cell senesces, then normally it will kind of start spitting out cytokines, chemokines, inflammatory signals. And there is some advantages to that in some cases. It's actually important for wound healing if a tissue is wounded. But once the cell becomes senescent and it starts spitting these things out, normally what that does is it triggers the immune response in your body to come and then get rid of that cell. Okay? If that starts to slow down, that getting rid of senescent cells, and it does with aging, what happens is you get an accumulation of these senescent cells, and they start putting out a lot of toxic things. And that leads to damage of the surrounding cells that maybe were happy before. Now they have DNA damage, and they go through the same thing. So this is one way, this is a mechanism, and it's known that cellular senescence is directly related to aging at the level of the organism. So in addition to what's going on here with apoptosis and cellular senescence, the other outcome that cell could have, and that's kind of where our lab is interested at the intersectionality of why a cell continues to divide versus become something worse, is if it escapes senescence, if it escapes that break, it actually can transform into a cancer cell. Okay, so that's the other outcome. So by comparison, this is better than this. So in addition to studying things at the cellular level, really in the context of aging, and wondering why are aging rates so different, and how does this relate with DNA damage and repair? There's been some really interesting studies on long-lived mammals compared to ones that don't live so long. So the longest living mammal is the bowhead whale. It lives over 200 years. If you look at the epithelial cells from bowhead whales and you look at how those cells repair double-stranded breaks, they actually repair it at a much higher efficiency than a human cell. People have taken those genes out and actually expressed them in a human cell in a petri dish, and they can extend the lifespan of human cells in a petri dish. So whether that translates, could translate into anything, interesting for us eventually, is to be determined. The naked mole rat, I don't know if you guys are familiar or have read anything about that. So that's a rat that's, it's a rodent. It's not much different. It's quite a bit different looking, but it's not much different from an evolutionary perspective than the rats, just like the rats around sewers and stuff. They live 10 times longer, so they can live up to 40 years by comparison. They have an increased efficiency of certain types of DNA repair. They also produce what's called hyaluronic acid. And so there's this comes in different flavors. There's high molecular weight, low molecular weight. Basically, it means that there's a lot of monomers put together, and so they're longer. And it's thought that these high molecular weight species somehow promote repair, support that environment. In addition to that, there's the NIH dog study. So I'm a dog lover, so before your minds go to, they're doing experiments on dogs, it's okay. These are companion animals, and they're actually really good to study because, one, there's different sizes, different shapes. They have, for how long they live, they have, from some certain breeds, they live quite a bit longer than others. In addition to that, they usually are pretty well fed, so it's not like they're having to worry about nutrition. They live in the same environments that we do. In addition to that, they also have access to health care. And so, they go for their checkups, just like we do. It's been shown in dogs that the ones that live longer, actually, that does correlate with their telomere length, so that connection is maintained. Also, the cancers that dogs get, they've sequenced those cancers, and they tend to show mutations in the same DNA damage repair factors as the cells that are mutated, or as the genes that are mutated in the cancers that, that are seen in humans. There's also been some GWAS studies, and so that's genome-wide association studies, where they looked at individuals that were over the age of 65 that didn't have one of the diseases that I described, and they asked what genes, variants are associated with that. And so what I mean by variant is I have a gene A and you have a gene A. Our gene A's might be slightly different. There might be like one little, A, T, G, or C that's a little bit different, but they still function. And what they found is that, a large number of these variants that were genome protective were observed in individuals over the age of 65. In addition to that, so this is a quote from Cher, 77. She was asked by someone, "Don't you think you're too old to still be into rock and roll?" And her answer was, "I don't know, you're going to have to check with Mick Jagger." So, actually their genomes would be really interesting to sequence, huh? Not for maybe the longevity, but the cool factor. In addition to, these GWAS studies, another group of humans that have really contributed to, and will contribute more, are centenarians. So that's the group of individuals that live over the age of 100. That group's growing, and they are usually the individuals that have managed to not get cancer, cardiovascular disease, or a neurological disease. That's been, warded off for a little bit longer. Those individuals, they've taken cells from that group, a group that's between 65 and I think 85, And then they also, the third group was younger tissue from younger individuals age 18 to 24. What they found was that centenarians actually had less overall DNA damage. They looked much more like the young group, 18 to 24. And when they were, challenged with oxidative stress, DNA was damaged, but they repaired it much more like the younger group. So they have this, the ability to protect their genome. So within the fountain of youth, this, because more people are living longer, this is really, there's lots of people who are trying to address this problem. And actually, the US Patent Office, in the last 10 years, the number of patents that have been filed have gone from 1 into the hundreds over the past 10 years. So, there's an interest not only in people who aren't trying to sell you something to make you live longer, but people, trying to also respond to that. Currently, there's nothing approved that can extend, that has been approved to extend human lifespan. There are things that have been shown to extend lifespan in the lab in model organisms, but these haven't been tested yet in humans. There are 50 clinical trials going on, and many of these trials take into consideration drugs that have already been FDA approved and repurposing them as, quote, longevity drugs. And two, maybe that you've heard of, one is rapamycin, and the other is metformin. So metformin has been used in type 2 diabetes, and rapamycin has been used in transplant recipients. So currently, actually, the best fountain of youth is, behavior and your choices. And a lot of that has, it correlates with what, maybe you guys have heard about these blue zones. There are regions in the world where they have a high number of people that, are living longer than in other places. And they have seen that some of the things that extend lifespan or keep people disease-free are exercise, so that's good, and circumstantial, I don't know any of the mechanisms, is actually, moderate exercise actually lowers your levels of DNA, underlying levels of DNA damage. Eating 80% of what you normally might. So this has kind of been a really interesting area of research, both caloric restriction and intermittent fasting. And it's thought that with this, you just have less that your like your cells metabolize. So there's less oxidative stress being produced. But intermittent fasting actually is interesting because it has this benefit, but it also has the benefit of, it's been shown that with intermittent fasting, what that leads to is your body reacts and it thinks, oh, I need nutrients. I don't know where my next meal is going to be. I better start breaking down some of the cells that aren't functioning properly. And the number one thing that they have seen targeted are those cells that are undergoing senescence, that are spitting out all these nasty things. So, that's the combination of getting rid of senescent cells and also not creating as much DNA damage. There's also an area of pharmaceuticals that actually target, and I think this is a really interesting area. They target senescent cells. They're called senolytics. And this is kind of a really interesting area of research because they target, the ones that are spitting out all these bad things. So yeah, but back to the blue zones. So good exercise, eat but not too much. Sleep. It's been shown that sleep actually is really good for DNA damage repair, especially your neurons. It's a time when your brain cells can go offline. DNA damage repair occurs faster when you're asleep in your neurons than when you're awake. And there are foods that are associated with upregulating repair enzymes. A few of those are broccoli, persimmons, lemons. But again, it's kind of circumstantial, and I don't want to give up my Christmas cookies for lemons. Anyway, so with that, I will stop talking about aging. And if you have any questions or would like a discussion about anything, we can start that.
All right, so I saw a few people kind of buying stock in the two drugs that Jen had advertised, so maybe a little premature. Are there any questions? Yes, please.
What is oxidative stress?
So the question is, what is oxidative stress? So just during normal cellular metabolism, so I'll give the example in the mitochondria, there's pathways that create ATP. That's the energy that your cell needs. The off products of that are reactive oxygens. So they're oxygen molecules that should be bound to something. If they're bound to hydrogen, 2 of them, it would be water. But they're an oxygen and they're missing something. And so they're highly reactive with things that are in the cell. And those reactive, they call them reactive oxygen because they can react. They create damage to DNA, proteins, and lipids. So many of the macromolecules that are found in your cells. But the contrast with DNA is the one macromolecule that can't be replenished. So, it's, again, the blueprint, and it codes for proteins or enzymes that are important for putting lipids together. And when proteins are disrupted, they can be targeted, which is another pathway that's disrupted in aging. But if things are functioning right, they can be targeted for degradation, and you can make more of it. If your DNA gets damaged, you can't make more of it.
Right. There was a question here. Yes?
What are you doing as far as stem cell transplants and gene therapy?
So the question is, what am I doing about stem cells and transplant therapy?
Fixed DNA?
So that's a good question. Our lab is personally not working in that area, so I probably don't know the latest. Stem cells are a big active area because as cells die, if they do undergo apoptosis and don't become senescent, then you need stem cells to divide again to make, like, let's say, the epithelial cells. And there's a limited capacity there too. So, and DNA damage actually occurs in stem cells. And when that happens, then you don't get anything else. You can't replenish. And so some therapeutics are targeting the stem cell niche.
Like you can do autologous stem cell therapy,
Yes.
where it's actually cured thalassemia in a couple of people.
Yeah. I don't know. I don't know what the latest is in the context of longevity, but I do know it's an area of active research.
There was a question in the back there.
Define intermittent fasting.
So, yeah, I guess that could mean something different to different people. So, intermittent fasting is what, and from the things I've listened to and read, it would be not eating or restricting your intake to within an 8-hour period of the day, and then not eating anything else around that time. And there's also some evidence to suggest that not eating right before bed is also good because it's like synergistic with restorative sleep, and not eating that it leads to doubly good things.
Do you live longer, or does it just feel longer?
Good question.
Actually, I should say, when Jen was talking about moderate exercise, I should point out she ran 5 Boston marathons. So I think we have to set the bar here at the right level. There's a question right here in front.
I listened to a podcast yesterday about artificial intelligence and longevity. Anything along those lines? The guy that was being interviewed at the end of the conversation, he was talking about all the publicly traded companies, and better jump on this one right now, and you better jump on this one right now because they're about to take off.
Oh.
I can tell you who the podcast was.
So the question was about the artificial intelligence and longevity. That is not my area of expertise. I do think artificial intelligence is going to be transformational for allowing hypothesis to be formed. So I think they're going to be very good at taking bits of information from, different areas and being able to assimilate it. And so I think they'll be good for forming a hypothesis, but then whether it's correct or not, I think scientists will have to prove this experimentally at the bench. And I don't know which companies they were talking about, but they probably have something to do with metabolism and inflammation and keeping those things down, I would maybe venture to guess.
Thanks. There's a question right there.
Broadly speaking, are there any known benefits associated with aging? So if we could take your magical genes out of naked mole rats or whales and put them into everyone, are there any known problems that we'd run into, or are there any hidden benefits that we don't know about?
So I think that's kind of a philosophical question, in that, I guess, the title of the talk was longevity and health span. We don't want to live if we're not healthy. So I think, most of where I see a lot of strides being made in the next few decades is to extend health span up to, the oldest individual on record is 122 years and I think 168 days. Jean Clement in southern France. And she did have a long health span. So, I guess there's benefit if you can live healthy for 122 years. You can, see more people in your life. I don't know if there's a benefit. Humans, once they get past their reproductive years, they don't have anything to offer the gene pool other than contributing to the lives of people that you care for. So I think there's benefit in that sense. Yeah.
All right, right here in front, and then in the back.
Yeah, thanks very much. I was wondering, my understanding is that restricting calories early in life is far more advantageous to extending your lifespan than doing later in life. Is there sort of an optimal period, or one to sort of caloric restrict, or is it beneficial at any stage in your life? It just may be more beneficial earlier on than later on.
it's beneficial at any stage in your life. I don't know enough about the age brackets on how much benefit it is. I do know that if you do one, 2 or 3 day fast once in your life, it can extend your lifespan by quite a few months, like maybe even a year. So, because it metabolizes these zombie cells, senescent cells.
Yes.
You talked about behaviors that will positively impact your health. Are there any, besides the inverse of those, are any specific behaviors that do the opposite, so that will accelerate DNA damage, like in particular?
Yeah.
And my other question is there any difference in what you talked about tonight between men and women?
Yeah, I mean, so on average, women tend to live a little bit longer than men. In terms of behaviors that, increase the incidence of these age-related diseases. I don't know, I think maybe early on cardiovascular disease impacts men more than women, but, women catch up. Different types of cancer, I mean, women obviously don't get prostate cancer. In terms of, I guess, behavior-wise or habits, it would just kind of be the inverse of, not getting good sleep, eating a lot of foods that are high caloric but low in nutrients, things like that. So, consuming, there's lots of studies that have looked at alcohol and, all of these blue zones, they were all associated with good moderate drinking levels, but there's a limit to that. So, and it's like
they're selling alcohol here, so
yeah, I know. I
kind of
should be
tone this
quiet.
down a bit.
Sorry. Probably one of the biggest things, at least for that we can do something about is sun, and making sure you wear sunscreen. That's a big one. They've done studies where they looked at truck drivers who spent so much time driving that the face that was towards the window, the skin on that side looked 20 years older than the skin on this side.
Wow, that's interesting.
Yeah.
There was a question right here. Yes, go ahead, please.
I was just curious, I had the pleasure of watching a documentary recently on the blue zones, and I wondered about sort of interdisciplinary research regarding stress. We know stress causes inflammation in the body, probably called cell senescence. What sort of research can you speak to the effects of stress and or the inverse of that, the benefits of relationships and low stress lives?
No, so that's a really good point, and that was definitely in the blue zones, and it's actually on my notes here, and I didn't say it. So having important social connections is longevity promoting, and it has been shown that lowering stress, maybe that's, through self-meditation or interactions with other individuals that you care about. It's known that telomeres are longer and actually persistent, in some cases, persistent levels of damage are down. But probably the biggest correlations are on telomeres, that telomeres don't shorten as quickly when you're less stressed. Actually, they did a study of mothers that had, experienced a lot of stress if they had children that were born with birth defects or some developmental defect. So when they were, trying to figure out what was going on under an enormous amount of stress, the telomeres in the cells derived from those mothers shortened at a much faster rate during that time.
There's a question here.
You
what the health span is.
Good question. So the question is, the lifespan is in the 80s, and what is the health span now? I think that is pretty subjective, and if I don't know the statistics on if you take an objective measure over 65, if you have a disease or not. And so that is the determining factor for health span. I don't exactly know what those statistics are, but living a quality life, that can be very subjective, right? So, it can be anything from I'm going to climb to base camp at 60 to I'm going to learn to play pickleball or a game of bridge. That part's subjective. But what isn't subjective is that people want to be disease-free and pain-free. And I don't know what the statistics are for people in that group over the age of 65.
Well, I ask because you always hear how the seniors are draining the healthcare system. And I was wondering, is there anything that says, now we're into it?
All I can say is I feel it. I feel you. I don't know.
There was a question right there. Sorry, I think you go first. Okay. Yeah,
Well,
sure.
kind of back to the telomeres. With CRISPR technology, is there any ongoing research that's inserting the gene for expressing telomerase to activate apoptosis?
So not so much. So the reason why telomeres shorten is not because telomerase isn't there. It's actually because it's not being expressed. So you have to be really careful with telomerase. And the reason why is because that's a mechanism that cancer cells use to survive. So if you start turning telomerase back on in cells that have acquired mutations, you might actually be causing cancer. And so there are some researchers out there who have done this where they've turned telomerase back on for a short period of time, like in mouse as a model system, and they could extend the life. I don't know the mutational burden that those animals, what it was relative to what it would have been. So there is research in that area. You just have to be really careful with telomerase.
There's a question right here in the back. Yes. Yep. Oh, yeah. Go ahead.
Besides eating good food, besides drinking water, besides doing exercise, what benefit toward the aging occurs with the practices of mindfulness and meditation?
So the question was, in addition to eating well, exercising, I'm going to throw sleeping in there. What are the benefits of mindfulness and meditation? I would answer that very similar to the question that was here earlier as a way to lower stress. It has been shown that individuals and monks that meditate have longer telomeres. So that would be preventative of cellular senescence. But I think in a way that too is subjective because maybe some people don't feel stress-free when they're meditating. I think the key is dealing with stress and what is good for that in your life.
Thank you. I think we have time for 2 more questions. Are there any? Yes, please.
Stress in early life, how can that affect?
Yeah, no, I think, so the question was, how can stress early in life affect the aging process? I don't know all of the literature on that. I guess I would, if I were to answer the question, I think it would be that there's a likelihood that you would be more predisposed to certain, age-related diseases. It doesn't mean you're going to get them. But there have been studies. Yeah, I don't know if any of you have read any of Gabor Mate's books and Stress and When the Body Says No and so forth. I would, I guess, point you in that direction rather than anything I'm saying. But I think he's demonstrated quite well that stress predisposes you to many types of health issues.
All right, one last question. Actually, you had a question before, right? I give you a second one.
Oh, sorry. Well, it just made me think of the idea of epigenetics.
Yeah.
And what sort of research related to epigenetics in cells, in essence.
Yeah, so that is a really interesting area of research. And epigenetics, so when I was saying the genome is packaged in a certain way and it's like highlighted in areas and maybe flagged, with a sticky note, that's kind of like the analogy of not the actual code, but how it's packaged. And that's where epigenetics comes in. So it means above the genome. But it impacts how the genome is expressed, behaves, and it has been shown by some researchers that after you have damage in a certain area and you repair, that the epigenetic signals in that area are different than they were before. So their idea was if you can reset the epigenetics, then you can reset longevity. And so what they did is they expressed some of the factors that have, and I don't want to get too technical here. So there are these factors, they're called the Yamanaka factors. And basically there are 4 transcription factors that basically can take a cell like an epithelial cell or a neuron and change it back to basically a stem cell. If they expressed 3 of those factors for a short period of time, they restored the epigenome and they were able to reverse aging in mice. What they don't know, and I think the jury's still out, is long-term, what is the mutational burden of those mice and their offspring.
All right. Well, I think absolutely fascinating, and I am really grateful that Jennifer could make the time. She's going to be here for more questions. Should you have any questions left, please join me in thanking her.
Thank you for coming out and listening to talk about aging.
Right. Thanks again.